This is part B of lecture CV2. In this part, we'll be talking about contractile versus autorhythmic cardiac muscle cells. We'll look at the anatomy of a contractile muscle cell, how its action potential and excitation-contraction coupling work, and then we'll look at how autorhythmic cell action potentials work and how they are influenced by the autonomic nervous system.
Cardiac muscle is unique to the heart. And in a way, it's sort of like a combination of skeletal and smooth muscle. It has some characteristics of both. There are actually two kinds of cardiac muscle cells. There are the contractile cells, which we sometimes call cardiac myocytes. And there are the autorhythmic cells. When you think about cardiac muscle, you're probably thinking of the contractile cells. Those are the cells that actually do most of the work of the contraction. The cells we talked about at the beginning of the muscular system, the branched, interconnected, striated cells, those are the contractile cells. Autorhythmic cells are a little different. There's a lot fewer of them. They are less than 1% of the heart. And they don't really participate much in the contraction. They have very little actin or myosin, so they don't really generate a lot of force. But instead, they're gathered into clusters and fibers in the heart. Their job is to generate action potentials at a steady rhythm and then spread those into the contractile cells, so they're starting the heartbeat, and then also to carry those action potentials into the right areas of the heart at the right time so they can coordinate which parts of the heart are contracting and in which order and for how long. So we're going to start by talking about how the contractile cells are put together and how their action potentials and excitation-contraction coupling works. And then we're going to move on to talking about the autorhythmic cells and how their action potentials work. Cardiac myocytes are striated very much like skeletal muscle cells. They've got the A and the I bands, which suggests that their actin and myosin is arranged in thick and thin filaments arranged in sarcomeres, just like in skeletal muscle. And that's true. And that means that when cardiac muscles contract, they contract in a way similar to skeletal muscle. They'll contract from end to end, unlike smooth muscle, which contracts all the way around. Now, in this cardiac muscle, these cells are joined to each other in this branched and interconnected network at these structures called intercalated discs. Those intercalated discs contain a number of interesting protein groups. So let's take a closer look at one of those. So here's a close-up view of two contractile cells where they meet at one of these intercalated discs. Now, one of the main kinds of proteins you'll find here are gap junctions. We've talked a little bit about gap junctions in single-unit smooth muscle. These are special channel proteins on both cells that link together to form a bridge from one cell to the next. Those gap junctions allow small molecules, like very small organic molecules, amino acids, but importantly here, things like ions, to flow directly from one cell to the next. So if you have an action potential propagating down along one cardiac contractile cell, the ions flowing in during that action potential can move right across to the next cell and trigger an action potential there, which will then continue to propagate. So for the purposes of things like electrical signals, these two cells are joined together quite tightly, and signals will spread directly from one cell to the next. The other kind of structure that you'll find here at these intercalated discs are groups of structural proteins called desmosomes. Desmosomes are sort of like cellular superglue. They're structural proteins on the surface that cause the two cells to link together very tightly. If you keep in mind, when these cardiac muscles are going to contract, when this cell pulls, it needs the other cells to come with it. We need the whole thing to work together to make this a usable muscle structure. But that actually leads us to an interesting problem. If this cell contracts, it's pulling this way, And when this cell contracts, it's pulling this way. If these were the only way in which these two cells were joined together, those two cells would tend to pull right apart. This would be a little bit like if me and my friend both put some super glue on our palms and stuck our palms together. When we both pulled apart, we could probably break that super glue. We might lose some skin. In the same way, when these two cells contracted, they could probably pull that apart. It might cause some damage to those cell membranes. In order to prevent that from happening, so that every time the heart beats, it doesn't shred itself into a mush, these desmosomes, color them in darker here to make it a little easier to see them, these desmosomes are attached to the cytoskeleton, the network of filaments and tubules that gives the cell its shape and structure. That way, when this cell pulls, it's not just pulling on those desmosomes, it's pulling on the entire structure of this cell and pulling it along with it. Now, my friend and I have not just put super glue in our hands, but that super glue is connected to the bones of my hand and wrist and arm, and my friend's, so that when I pull, or when she pulls on me, it's pulling on my entire skeleton. Nothing is going to rip free. It's going to pull me along very effectively. I always hate kind of looking at this diagram because it reminds me of these. If anyone's lived in the Midwest, they're familiar with daddy long legs, also known as harvestmen. Maybe we'll have a quick thing on here about those, just for the sheer joy of it. So from that, what you can imagine is that my contractile cells in the heart form an interconnected network so that an action potential in one of them spreads through the network, causing each cell to twitch as we're going. So what we get is a wave of contraction spreading over the whole area of connected muscle, which is a little bit like single unit smooth muscle that we talked about in a previous part of the class. Okay, now let's talk about cardiac contractile cell action potentials. These are similar to the action potentials we had in nerve cells up to a point, but then they differ pretty dramatically. So to begin, let me pop up here a chart that'll let me show membrane potential over time. Good. Now, let's take a look at how this will work. Let's see, what color can I use here? Okay, so my action potential in cardiac contractile cells is going to involve starting at resting membrane potential, which is down pretty close to negative 90 in these cells, a little more negative than in nerve cells. And as we talked about in nerve cells, something is going to push me up to threshold, which in these cells is about negative 70. That's when the voltage-gated sodium channels are going to open. So when I reach that threshold, I trigger the opening of the voltage-gated sodium channels, which, like in a nerve cell action potential, causes a rapid depolarization up to pretty close to about positive 50 or 60. Now, when I reach that point, those sodium channels go from open to inactivated, very similarly to how they do in nerve cell axons. And we get some potassium channels opening, which causes me to begin to repolarize. So far this is very much like a nerve cell action potential, but then something interesting happens. About the time I get to roughly positive 30 millivolts, I have another channel type open. Calcium channels. And this is going to have calcium flowing in. Now, positive calcium flowing in tends to make the inside more positive, depolarizing the cell. So that's now sort of fighting against the potassium, and I end up in this plateau where the membrane potential stays positive for quite a while because of calcium inflow and potassium outflow. that lasts for roughly 150 to 200 milliseconds. And at the end of that, the calcium channels close. And potassium is then going to bring the cell membrane potential back down toward my resting membrane potential. So that's where my calcium channels close. So if we were to just kind of sketch in what's going on here, this part is due to sodium inflow. Here's potassium outflow through this whole period. And then I have calcium inflow during this period. That potassium is meant to be continuous through there. Although this is slightly simplified. There's actually more than one kind of potassium channel contributing to this, which open and close at different times. But what we're getting at here is that we have this extended plateau of about the whole thing from beginning to end is 200 to 250 milliseconds. So we've got this action potential is unusual in that it stays depolarized for a long time, much longer than a typical, like say, nerve action potential, which is over in about a millisecond. Now, there is something interesting to note about that. These sodium channels, which inactivated up here, you'll note that the sodium channels inactivate here. Remember, sodium channels have to get down below a certain voltage in order to go back to their regular closed state. And that's not going to happen until pretty much the end of this action potential. So here's where the sodium channels go to their regular closed, not inactivated. So this is when they're open, inactivated, and then closed. Now remember, the absolute refractory period, the time when a cell can't have another action potential, is when the sodium channels are inactivated. So in this cell, that absolute refractory period lasts pretty much from here all the way to here. During that time, this cell cannot have another action potential. Now let's add something else interesting onto this. If I were to add down here the twitch force, this cell starts twitching just a few milliseconds after the action potential begins. And that twitch lasts just about as long as the action potential. So my cardiac muscle twitch is roughly the same length as that action potential. So think about this. If my sodium channels inactivate here and don't become available again until here, when that twitch is effectively done, how could I get summation in a cardiac muscle contractile cell? How would I be able to add another twitch onto this? Take a moment to think about that. Okay, you may remember that we had a challenge question that was a little bit like this. It was saying what would happen if the absolute refractory period in a muscle cell was as long as the twitch. Well, here we have that. And if you remember, the answer to that challenge question was, if the action potential is as long as the twitch, you can't get summation. That's the case here too. This cardiac contractile cell cannot summate twitches because the soonest it could start another twitch is here when the first one is done. Then the other question is, why would a cardiac muscle cell ever want to summate twitches? The whole point of the heart is to contract and then relax. If you imagine a cardiac muscle cell summating twitches into some sort of tetanus, it would go and stay contracted. And we never ever want the heart to do that. So really, this is a safety feature. No matter what kind of stimulation you give to cardiac muscle, you can't get those muscle cells to seize up in a tetanic contraction. Because of this plateau, they force themselves to relax before they'll twitch again, which is kind of cool. Okay, so let's talk about the contractile process in these cardiac muscle contractile cells. Now, in many ways, it's very similar to skeletal muscle. Cardiac muscle cells have troponin and tropomyosin in their thin filaments, in their myofibrils. They have terminal cisterns, which have calcium in them. So, as before, calcium is going to bind to troponin. Troponin is going to move tropomyosin. Myosin can bind to actin, and the myosin will go through its contractile cycle. That part is very similar. The contractile cycle is effectively identical. It's kind of like slow twitch skeletal muscle, but even more so. Remember, the cardiac muscle twitch is more 200 to 250 milliseconds, which is even slower than skeletal muscle slow twitch, which is about 100 milliseconds. But let's talk about one thing that is a little different, which is excitation-contraction coupling in cardiac muscle. So let's start by sketching in our cardiac muscle cell. And it's got T-tubules and sarcoplasmic reticulum with terminal cisterns. So far, very much like our skeletal muscle. We have calcium pumps, calcium ATPases on the sarcoplasmic reticulum, which are making sure to pump calcium from the sarcoplasm into here. So my calcium builds up in here. So far, much like skeletal muscle. And I do find DHP receptors on the T-tubules and ryanodine receptors on the terminal cisterns. We'll get to those in a second. There is something a little different there. But I do want to point out one other difference that you'll find here in cardiac muscle from skeletal muscle. In cardiac muscle, we also have calcium ATPases, calcium pumps, on the sarcolemma. So those are pumping calcium out here, so we have calcium outside the cell, which includes here in the T-tubules. That's going to be important here in just a moment. So what I want to do is blow this up so we get a closer look right here at what's going on where this DHP and ryanodine receptor are. Okay, so here's our close-up version. You can see our T-tubule with calcium inside it, DHP receptor here, ryanodine receptor here on the terminal cistern, which also has calcium in it. If we want, we can sketch in those calcium pumps just to keep things consistent. Now, one thing here is very different from what we saw in skeletal muscle. Do you see it? In skeletal muscle, the DHP receptor was physically attached to the ryanodine receptor. Remember, we did the thing where the action potential activated the DHP receptor, which twisted and pulled open the ryanodine receptor, releasing the calcium. That was how, in skeletal muscle, we got calcium release in response to an action potential. In cardiac muscle, there's an interesting difference. They are not connected. The DHP receptor is not physically attached to the ryanodine receptor, which seems to lead us to a problem. If that's the case, how are we going to open that ryanodine receptor? Well, there's another difference that's a little harder to see. You may remember in skeletal muscle we mentioned that the DHP receptor, while technically it was a voltage-gated calcium channel, it didn't actually work as a calcium channel. In cardiac muscle, it does. See, there was a reason I made sure to make a big deal of that. So here, this is a fully functional voltage-gated calcium channel, which means that when an action, here we can do this as a sequence, when action potential propagates into the T-tubule, so here's my action potential coming down. That should really be with the plateau, I suppose. The action potential causes the DHP to open. So my action potential opens my DHP receptor. And calcium can enter from the T-tubule. This is why it was important that we had calcium out here, one of the reasons. Now, how is that going to help us? If you remember, the ryanodine receptor is a calcium channel too, but it is also gated by calcium. So when this calcium comes in through the DHP receptor, that calcium will bind to the ryanodine receptor, causing it to open. So calcium enters through the DHP receptor. Calcium binds to the ranitidine receptor. The ranitidine receptor opens and releases calcium from the terminal cistern. So out comes my calcium, a big flood of calcium from there. That calcium will be a lot of what gets to my myofibrils, binds to the troponin, moves the tropomyosin, etc. Another thing that affects this is, remember those calcium channels opening during the action potential, letting calcium in from the inside, which caused the plateau? That calcium coming in during the action potential also can bind to troponin, so it sort of amplifies that. This doesn't have to do all the work. So this is one source of calcium, another is coming in during the action potential. That calcium then gets to the troponin. So a little bit different, but that's how excitation-contraction coupling works in cardiac contractile cells. All right, so now we've learned all about how cardiac contractile cells work, how they're physically arranged, how they connect to each other, how action potentials can propagate from one to another through those gap junctions. We learned how their action potentials work, and we learned about how that action potential leads to calcium release from both the terminal cisterns and calcium coming in from outside during the action potential. And that calcium binds to troponin, you know the drill from there. So we know a little bit about how the cardiac muscle works, but what we don't know yet is, where does the signal come from to start this whole process? We mentioned that the cardiac muscle works by intrinsic activity, that it doesn't have to be told to contract by something from outside. If you remove the heart from the body but keep it alive, it'll continue to beat. So where does that signal come from? We mentioned that there's another kind of cardiac muscle cell called an autorhythmic cell. Those autorhythmic cells don't really contribute to the contraction, and they're rare, only about 1% of the muscle. But they're arranged in little nodes and fibers, and their job is to create the action potentials that start this, as well as to carry them to the right places in the heart to coordinate the heartbeat and make sure different areas contract at different times. We're going to get more into how that's coordinated in our next lecture, but right now, what I want to talk about is how those autorhythmic cells make those action potentials, because they work a little differently than they do in the cardiac contractile cells. Okay, so let's start again with a graph of membrane potential over time, and here we're going to be looking at an autorhythmic cell's action potential. These cells don't have a very well-defined resting membrane potential because there really isn't any one voltage they tend to go back to. You'll see why in a moment. But we're going to start at sort of the low point in their cycle, when they're starting out down around -75. At that point, these cells are on their own slowly depolarizing without anything else causing that to happen. Now, I can tell you right now that when people were first studying these, they noticed that these cells, after their action potential, started to depolarize themselves back up to threshold. They looked at the membranes and saw there was an inward flow of positive ions, in this case sodium, which was causing that depolarization. And they said, that's funny. And because scientists are the way they are, they decided to call the channels involved in this, letting this sodium flow in, funny channels. Because they're scientists. In any case, these funny channels allow the cell to depolarize, they cause the cell to depolarize up until it gets to around -40. And just before that, we actually close the funny channels, but bring in a little bit of calcium flow, which brings us up the rest of the way to that -40. When we hit -40, a different set of calcium channels opens, and we get a depolarization due to calcium inflow. This is a little different from the action potentials you've learned about so far. The spike type thing here comes from calcium rather than sodium. It's different channels than those calcium, so we'll put it here in a dotted line to show that it's a different kind of calcium channel. So calcium flow causes us to depolarize for a little while. That's going to bring us up to somewhere around, again, maybe about positive 30. When that happens, the calcium channels close. But potassium channels open. So here it's a little bit like our action potential of the type we've already learned about. When those potassium channels open, that's going to cause the cell to drop back down toward this negative 75-ish range. Let's indicate that that's because of potassium outflow. When we get down toward negative 75, pretty soon after that the potassium channels will close. But the interesting thing that happens there is rather than staying there at resting membrane potential, that's where these funny channels come in. So the funny channels open at around negative 75 and will start bringing us back up toward threshold. So here's where the funny channels open. Which will bring us back up toward threshold when we get close. Even though the funny channels close, we get some calcium channels open that bring us up the rest of the way, and we start another action potential. So this is an ongoing cycle. We have an action potential, which causes the funny channels to open, which depolarize us back up toward threshold, which causes another action potential. So we get action potentials in a regular sequence. Thus the idea of autorhythmic cells. So that's all well and good. But one thing we know about the heart is that it can speed up and slow down based on input from other things. So while the heart controls its own heartbeat and initiates its own contractions by these autorhythmic cells starting action potentials on their own, we know that it can be influenced by, for example, these sympathetic and parasympathetic nervous systems. So somehow those must be able to influence how quickly we have these action potentials. Let's take a look at how that works. So I'm going to erase some of this so we've got a little more room to work with. Okay, first let's talk about what happens when we have more sympathetic input. So when the sympathetic nervous system is more active, our sympathetic postganglionic neurons are releasing norepinephrine onto the beta-1 receptors on these cells. Remember, we talked about how among the adrenergic receptors, beta-1 receptors are found on the heart. They respond either to norepinephrine or epinephrine. In this case, we're looking at norepinephrine. And they cause an increase in activity. Here's what that means in these cells. When that norepinephrine acts on beta-1 receptors, what those do is act to open more funny channels. So under that influence, rather than this relatively slow depolarization back toward threshold, instead, under sympathetic input, I'll depolarize faster because I've got more funny channels open. So activating the beta-1 receptors causes us to open more funny channels, which brings us up to threshold faster, which means I start my next action potential that much faster. And you can see that rather than having this long period between heartbeats, like say if this was 1 second, maybe now it's happening every 0.6 seconds. This would be the difference between a heart rate of 60 and a heart rate of 90 beats per minute. So the sympathetic nervous system can speed up the heart rate just by making us depolarize back to threshold faster. All right, hopefully that makes some sense. But what about the parasympathetic nervous system? If the sympathetic speeds it up, The parasympathetic should be able to slow it down, and it does. For parasympathetic, we've got acetylcholine acting on M2 muscarinic receptors on these cells. Those M2 muscarinic receptors cause us to keep the potassium channels open longer. So those potassium channels that were open to bring us down here, under the influence of M2 receptors, they stay open longer. When those potassium channels are open longer, that's going to slow down the depolarization. That acts against the funny channels. So it's going to take me longer to get back up to threshold. And my action potential will happen later. They'll be farther apart, which means a slower heart rate under parasympathetic stimulation. Now, it's also worth noting that those can both be happening at the same time. We can have the sympathetic nervous system opening more funny channels and the parasympathetic nervous system keeping the potassium channels open longer. What that tends to do is sort of cancel each other out, and that's actually the normal case. Remember, for most organs, including the heart, the normal situation is to have levels of sympathetic and parasympathetic stimulation which are both there, just going back and forth. So the question is, who's got a stronger effect than the other? I hope that makes sense. In the last part of this lecture series, in CV-2c, we will be talking about how these autorhythmic cells conduct the action potentials through the heart and make sure that we have the heart rate happen at the right time and the right place and in the chambers contracting in the right order. So we'll come to that soon.